So here I want to tell you about the metric of color space. And all what I'm going to tell you here is part of the PhD thesis of Nicolas Rattuone that will be defended actually next month here in Instituto Balseiro. Okay, so let's start with the main question. What I want to ask whether there could be a particular reference system, reference frame, and geometry with which color space should be represented. Here I show you two examples of color space. There are many, and the transformation between one color space and the other are typically nonlinear. But then one could ask, is there a better one? Is there one that we could call the best one? And in order to think about this question, I would like to invite you to think about the very same question that we could ask for physical space. So is there a coordinate system in which position is best represented? And the answer to this question is yes. I mean, in physics, we know that there are specific reference frames, namely those that are inertial and Cartesian, in which physics appears to be maximally symmetric, or, if you want, particularly simple. What would that mean? For example, what would happen if you were to use a weird reference frame for position? Things like these would happen. So imagine that we are seeing the physics that is reflected in a distorting mirror. Now, if you in the real world move with a uniform motion, in the mirror, you're going to see some sort of jerky movement and some crazy accelerations that do not actually reflect properties of the real world. They reflect properties of the mirror. So in a way, what you're seeing are properties of the lens through which you have chosen to represent position. And we don't want that. In particular, we don't want it for color. So the question we want to ask is whether there exists a reference frame in color space in which perceptual phenomena are minimally contaminated by the weirdness of the frame itself. Or in other words, is there a frame in which perceptual phenomena are maximally symmetric? So this is what we want to do now. We want to search for a reference frame, a system of coordinates, and a geometry for color space in which perceptual phenomena such as, for example, just noticeable differences, or another example, induction phenomena, appear to be maximally symmetric. And the plan is as follows. We're going to measure these so-called just noticeable differences that I will explain shortly, and we're going to use those experimental results to define a coordinate system that makes those experimental results symmetric, in particular isotropic and homogeneous. And then in those coordinates, we're going to represent the results of induction phenomena, induction experiments that have not been used to construct the reference frame and to check whether their induction phenomena appears to be homogeneous and isotropic. So to do that, we have to start by the hypothesis that color space can be described by a Riemannian geometry. A Riemannian geometry is one in which if you select a point in space and then you select all the set of points that are at a fixed distance from the selected point, you get the surface of an ellipsoid. This is a Riemannian geometry. One particular case of a Riemannian geometry, the one that is most symmetric, is an Euclidean geometry, in which all ellipsoids turn into spheres and all spheres have the same size. So in principle, a Riemannian geometry may contain ellipsoids that have different sizes and orientations and eccentricities, but then it may or may not be the case that a coordinate transformation exists that takes the ellipsoids into spheres. Now, if we want to employ this for color space, we should first check that in fact, if you select a color and then select all the colors that lie at a fixed distance from the selected one, conform an ellipsoid, right? To do that, you first have to define what you mean by all the colors that lie at a specific distance. We don't want this distance to be some sort of capricious choice of a mathematician. We want the distance to represent something that has to do with perception. And that is where we put in the just noticeable differences that I mentioned before. So the just noticeable differences will be something like the minimal distance that we can detect perceptually. And this is how you measure it. You choose a color, and then you start to modify it until you can perceive the modification. You can see, I see it different. Okay? Since color space has 3 dimensions, this modification can be done in 3 different independent directions in color space. So you can start from the initial color and then start to make it slightly more bluish or less bluish, or more reddish or less reddish, and so forth. In principle, you can make it in infinite directions in color space, but only 3 of them should be independent, right? Now, people have done that, and people have checked that indeed the surfaces are ellipsoids, right? We have replicated those experiments, and for each observer, we have measured the ellipsoids, and we have checked that indeed a coordinate transformation exists that can take all the ellipsoids to be Euclidean. So we transform all the irregular ellipsoids, so to speak, into regular spheres. And that we do for each observer because the ellipsoids of different observers, although they have some general properties, they're not all the same. There are significant differences between observers. And that is something that we learn from our experiments. But this is the situation right now. We have a set of coordinates for each observer where all just noticeable differences are spheres, right? And now we want to see whether induction phenomena, which is the other experiment that we performed, looks symmetric in those coordinates. These coordinates, these preferred coordinates, we're going to call perceptual coordinates because they were obtained with perceptual experiments of just noticeable differences. So first I have to explain what induction is, what chromatic induction is. And chromatic induction is in short, is a perceptual change in the perceived color of a stimulus due to its background, okay? So here you can see 3 colored stripes, and in each stripe you can see 4 squares. Now what I'm going to tell you is that all the squares that belong to the same column have the same chromaticity, so the same electromagnetic spectra, so to speak. Now, if you look at them carefully, you will see that they look differently, right? So let's focus, for example, on the 3 squares that are on the first column. All the 3 have the same chromaticity, but the upper one, at least to me, appears to be more yellowish. And the one in the middle and below, they appear to be a bit more pinkish. And that is the effect of different backgrounds, right? In the second column, for example, I would say that the upper square is kind of more bluish and the second is a bit more reddish. And again, that's the effect of the background. Now, if you want to get some general idea of what backgrounds produce, what effects they induce on perception, essentially the effect of the surround is repulsive. So they shift the color that you perceive in a stimulus in order to make it more different from the surround. And we could argue that this has evolved in order to make chromatic borders more easily detectable. So what do we mean by backgrounds being repulsive? For example, here we can depict this effect as a repulsive field. The position of the white dot marks the color of the background, and each of the arrows tell you how a color is modified due to that particular background. For example, let's take one arrow. The base of the arrow is the color that you would perceive if the color were uniform in space, and the tip of the arrow is the color that you actually perceive the same stimulus when surrounded by the color indicated by the white dot. Okay? Now, what would be induction to be symmetric? It would mean that the repulsive field has to be spherically symmetric, right? And also that it has to be homogeneous. That means if you change the color of the background, you should rigidly shift the whole repulsive field, but with making no other distortions than that. Mathematically, we can write this down in a specific way. I'm going to say that S prime, which is the perceived stimulus, and that would be the tip of the arrow, is a given transformation T of the real stimulus, so to speak, that would be the base of the arrow, S, and the background, B, that's located at the position of the white dot. Now, the transformation T under the symmetry hypothesis is not any arbitrary transformation, but is a very specific one, at least in the perceptual coordinates that we assume to be the symmetric ones. The stimulus S prime has to be equal to the original stimulus S, but modified in a direction given by the vector R, which has to be a radial vector in the direction connecting the background and the stimulus, and by an amount that only depends on the distance between the background and the stimulus and nothing else. Okay? So this hypothesis has several predictions, and we have checked several of them, but today I'm going to tell you just one of them due to lack of time. Okay? And what I'm going to test is the following prediction. Chromatic induction varies when the surround changes, right? Because you have to shift the repulsive field. But the shift produced by a background remains constant if the distance of a stimulus to its surround remains constant. So what do we mean by this and how do we test it? We're going to take a uniform color B and a uniform color B prime, and the two of them are going to act as backgrounds. And inside of them, we're going to put a stimulus S and a stimulus S prime as in the figure, right? S is fixed. Now, an observer is asked to change S prime until he or she sees it exactly equal to S. And when they are happy, they give you the color S prime. And you, as an experimentalist, will see that S prime has not the same chromaticity as X. The electromagnetic spectra are different. And why is that? Because the observer is trying to compensate the effect produced by the background. Now, the shift is the difference between S and S prime, and that would be equivalent to the length of the arrow of the repulsive field that I've shown before. That the length of that arrow under the symmetry hypothesis may depend on B prime, on S prime, and on the direction in color space that we are exploring, on whatsoever. Many things. But if the distance between B and S and the distance between B prime and S prime, both of them remain constant, then the length of the arrow should be always constant. We should be traversing the repulsive field always within the same distances. Okay? So we have checked that that is indeed the case. And this is what I show in the graph. We have shown, here I show you the results of the experiment in the perceptual coordinates and in another coordinate system, the cone contrast, but we have checked in many other coordinate systems. Now, the length of the arrow is the vertical axis, the zeta axis, so to speak. And the other two axes depict the distances between B and S on the one hand, and B prime and S prime on the other hand. And you can see there two sheets, and those sheets represent the experimental results when exploring different directions in color space and different initial stimuli, different values of B prime, S prime, whatsoever, right? And we see that in the perceptual coordinates, all the data plots converge into a two-dimensional sheet. In the other coordinate system, instead, you have one sheet that is big and blue and the other sheet that is way below and much smaller, and actually the dimensionality of that collection of points is not 2, it's actually 3. It's more similar to a cloud of points than a sheet of points, which means that it's not true in the cone contrast coordinate system that the length of the arrow only depends on these two quantities. It does not have these symmetries, so to speak. So that is one of the checks we have made. There are more, but there's no real time to tell them all. So just to conclude, we observed that there is indeed a privileged family of coordinate systems in color space, and in the perceptual coordinates, which are the privileged, thresholds and induction are isotropic and homogeneous. They are maximally symmetric, if you want. And we also want to mention that there are significant differences between different observers. So we need to construct the coordinate system for each observer. More details you can find in the paper Vattuone, Wachtler, and Samengo, and it has been published in it's the first paper of the first number of a new journal. It's free access, which is called Mathematical Neurosciences and Applications. Okay? So just to I thank you for your time, and I also want to thank all these institutions that have made this research possible.
Okay. Oh, hello everyone. Again, we have some troubles at the beginning, so let me introduce again to Professor Inés Zamengo from Argentina. Dr. Zamengo got her master and PhD degrees in physics from the Instituto Balseiro, Universidad Nacional de Cuyo in Argentina. Currently, she is adjunct professor at Instituto Balseiro at the same institution and el Departamento de Física Médica y Centro Atómico de Bariloche at the same country. And she's also independent research at CONICET. Her group has been working, as you can see in your pretty nice talk, in the development of theoretical methods that help us to characterize the neural code that the brain employs. Thank you so much, Ines, for your very nice talk. I see. Remember that you can leave your questions in the chat for Dr. Zamengo. So we have room for one question. I'm just wondering, Ines, what about the perception in people that is lacking of one channel of color, just like me. I'm daltonic, and
Okay.
yeah, I am. So how your model behave in this case?
Actually, what I have told you today, we built the metric of color space by first measuring the discrimination ellipsoids of specific observers, and we should do that with you and other colorblind people or daltonic people or whatever, which is 1 in 10. I mean, it's quite a common condition, so you shouldn't feel bad about it. We also have a theoretical model to predict that. I mean, you can also make a model at the level of photoreceptors and come up with an equation of how the ellipsoids would be for people with different numbers of receptors, in your case lacking one type, but it could be also animals with more than 3 receptors, so you can construct them. I have to confess anyway that experimental results, I mean those models fit pretty well the experimental data up to 87% of the variance we explained, so it's pretty good. But it's better if you actually do the experiment person by person, because we have to say that there are individual differences. I mean, color blindness is typically not just one type of cone lacking, but it could also be one type of cone a bit weird, so its physiology is not functioning exactly the same as other people, and maybe the numbers of photoreceptors of each type vary from person to person. So it's something that you can describe theoretically up to a certain degree with models, and then if you want to go further, you have to do the experiment for each observer.
Yeah, I totally agree. The perception is really different in this kind of people. I'm really happy to do the experiment if you want later.
Yes, but okay, it would be great. By the way, people with different number of receptors, it's not so much how they perceive pure color, say the rainbow, that changes, but most of all how they perceive mixtures, because it's mixtures that vary. I mean, if you see the metric of color space, you see that on the line representing the one-dimensional locus that corresponds to monochromatic stimuli, okay, you see some small differences. But on the bulk of color space, I mean, in the middle of the three-dimensional construction of color space, which colorblind people typically have two-dimensional, there you see many more differences. Yeah. And I'm very jealous of the people who have 4 types of photoreceptors. I mean, women and very few. But I'm really jealous. I mean, I would love to know how that feels.
Yeah, me too. That's really, really interesting. Let's move on. We can go back to this or another question in the final remarks. Thank you so much, Ines.
Thanks to you.
Nice. I want to introduce our second speaker today is Professor Antonia Marin Burgin. also from Argentina. Dr. Marín Burgin got her PhD in biological science from the Universidad de Buenos Aires. She is currently leader on the neuronal circuit group from the Instituto de Investigación en Biomedicina de Buenos Aires, IBIOBA, CONICET, which is partner institute of the Max Planck Society, an independent research of CONICET, Argentina. Her group has been interested to understand how neural circuits code for sensory stimuli and how the experience shape this coding process. Her talk is entitled Experience-Dependent Changes of Sensory Representation in the Olfactory Cortex. And thank you so much, Professor Antonia Marín Burgin, for being here today. So the screen is all yours.
Hi, everybody. I would like to first thank the organizers for inviting me to this symposium and to tell you about our project on experience-dependent changes of sensory representation in the olfactory cortex that we are doing at the IBIOBA. Okay, let me start. Okay, here we go. So the project is related with the idea that when an animal encounters a stimuli, depending on which context and which experience has been associated with the stimuli, the same stimulus could produce very different behaviors. For example, in this case, the mouse can smell and can also see the food. And if he's hungry enough, he would probably go and approach the food to eat. However, if that particular stimulus was previously perhaps associated with another guest that was the owner of that food, maybe now the mouse, when he sees the stimulus in a subsequent presentation, would actually evoke a very different behavior. He would escape this time. So that is to give you the idea that perception is really not a passive action, but it also is shaped by the recipient's learning, memory, expectation, and attention. And what we would like to learn or to study in the lab is how experience modifies the representation of sensory information and where does it modify it in the brain. So to start answering this question, we chose the olfactory system to do that, and in particular the olfactory cortex when animals are performing a sensory motor task. And just a very brief introduction to the olfactory system. So the olfactory system is composed first, the other molecules arrive to the nose, to the olfactory epithelium, where a particular other molecule would activate one type of olfactory receptor neuron, and the same type of olfactory receptor neurons converge into a particular glomerulus in the olfactory bulb. And from there, information travels up to the brain, to the piriform cortex, which is the main region of the primary olfactory cortex. And the first question that we had, or the first idea to approach this question, was whether neurons that represent in the olfactory cortex a particular odor or the population of neurons, when the odor is presented in a context A, for example, that is associated with reward, would be the same population as when the odor is presented in a different context that perhaps is not associated with a reward. Are these two populations of neurons representing the same odor, the same, or different? And we believe that it might be different because of the nature of the inputs that the piriform cortex receives. So not only it receives information about the odor itself from the olfactory bulb, but in addition, it receives information for higher order processing regions in the brain, for example, the hippocampus through the entorhinal cortex that could confer contextual information to the odor representation, or for example, from the amygdala that could confer hedonic balance to the odor representation, as well as from other higher order processing regions like the prefrontal cortex, the orbitofrontal cortex, perineal cortex. So with that, we wanted to develop a task that would allow us to compare the representation of odors in different contextual, visual contextual scenarios, and also associated these contextual, visual contextual scenarios with the reward. And for that, we have a very, very good team in the lab. These two investigators from the lab, Noel Federman and Sebastián Romano, are really the driving force of this project. So we develop a behavioral task in which animals are head fixed. They but they can run in a running wheel that moves a virtual reality. And with this virtual reality, we can present a context, two different contexts. And they are a little thirsty, so they learn to lick or not, depending on the combination of an odor that we deliver with the context. We can also record acutely the neuronal activity in the piriform cortex. So as the animal move, they have to move in this corridor. As when they move with the running wheel, they can move the virtual reality. I will show you a video on that. So they enter the corridor, they approach either context A, which will be a green context, or context B, and they receive an odor as soon as they arrive to the context, and they have to, depending on the combination of odor and context, they have to lick or not for the drop of water. So there are 4 types of trials, 2 contexts and 2 odors, and only context A, odor 1 would receive a reward, and the animals have to learn to lick in this combination of context and odor, and to not lick in the other 3. And we also measure not only the licking, of course, response of the animals to the different trials, but also the inhalation rate, the speed, and the position along the corridor. And so this is a video to show you how the behavior looks like. So this is the mouse running in the wheel. It's moving the virtual reality. Now the gray context is coming. It enters, receives the odor. This combination is not predicting reward, so it will run through. And then he's approaching now the green context, and it will receive an odor. And then depending on the combination, now he will decide whether to stop or not to lick for water at the end of the context. So Macarena Amigo Duran did her undergrad thesis together with Noel Federman in the lab, and they trained these animals, and the animals learn pretty quick this task. They learn to only lick at the rewarded combination, and they learn in around almost 6 days. They become almost experts. So the D prime, the animal performance of 3.2 corresponds to around 90% of correct responses. And what we wanted now to do is to compare the activity of the neurons in the piriform cortex in these 2 different conditions, when animals are fast put in the behavioral task, or when animals are experts in the task. And so to record, so what happens in the brain, that's what we are interested in, and in particular what happens in the piriform cortex. And so to record from many neurons at a time, we're using silicon probes. We're using 32-channel probes, and we can record action potential extracellularly from the piriform cortex. So we label the probes with a dye, so to make sure that we're recording in the piriform cortex. And we can use algorithms to separate spike sorting. So we can separate neurons, and we obtain around 20 neurons per animal on average. So I'm showing you here an example of one particular neuron. You can see here are around 75 trials of this neuron, and then each of these lines is an action potential of the neuron. And down here you can see the inhalation rate of this animal, and we align the spiking activity to the inhalation rate. This green area is when we deliver the odor, and T0 is when is the first inhalation after the odor presentation. You can see here that this neuron responds almost with each inhalation to the odor presentation, and then it habituates. And so this is a typical odor neuron. We are in the piriform cortex, which is an odor processing area. So having odor neurons there is good, but it's not so exciting. So this is not the only thing we see. So this is another example of another neuron. You can see that this is another 2 cell. So it will increase its firing rate with other two, regardless of the context in this case. So this will be not responding to other one, but only responding to other two in context green and in context gray. But in addition to seeing other cells, we also see some cells that are very interesting. For example, we are seeing this neuron, and this is not here, this is not time, but this is position along the corridor. If we align the responses to the position along the corridor, and this represents the context position, so we can see that the firing rate of this particular neuron increases as the animal approaches only to context green, so to context A, but not when it approaches context B. So this will be a context cell, and we also found cells that respond to the rewarded combination. So in this case, this particular neuron increased its firing rate even before receiving the reward. So it's anticipating that there's going to be a reward at the end of this context corridor. So this is really nice, and we are not just guessing what the neurons are responding to, but we are actually now using generalized linear models to classify the neurons and which of the behavioral variables modulate their activity. And we are finding that not only, of course, others modulate the activity of neurons, but also position, context, reward, leaking, speed, and inhalation, and combinations of those variables are modulating their activity. So this is really nice because it means that the piriform cortex is not only representing the others, but it's also representing the whole behavioral task. Can we visualize that? And so to visualize that, we are using principal component analysis to actually look at the population activity of all the neurons that we're recording in a particular animal. So in this example, I am showing you here that as the animal moves through position, the activity of the populations of neurons in the 4 types of trials is moving similarly at the beginning. So at the beginning, the animal enters the corridor, and the activity of all neurons in the 4 types of trials is very similar. And then as the animal arrives to the context, you can see that it separates. So trials that the activity in the neurons of trials that are in the green context separate from the trials that are in the gray context. And as the animal received finally the odor, and that will actually be an association with a reward that is coming, the rewarded trials, or the activity of the neurons in the rewarded trials separates from the one that are not going to receive a reward, that it almost looks like it's going back to the similar to the gray context that is not rewarded. So that means that by looking at the activity of the population of neurons responding to the task, you can actually see differences in the type of trials that the animal is experiencing. So the next question we had is whether is this like this from the beginning, or is it actually something that is acquired with learning? And for that, so this is of course an example of an expert animal, but if we compare this with animals that have been just put in the setup, we can see that this is not the case. So actually the activity in the of the population of neurons in the different types of trial is all mixed together, and you cannot really separate the trial type in the task, because the animal doesn't, for the animal it doesn't mean anything, he's just there, he has to learn the associations, and the activity of the neurons represent that. So that means that it's actually the encoding of the different variable tasks related, the different behavioral variables related to the task is actually generated by experience. And to further see or to further analyze which are the variables that are actually acquired with experience. See, so what are the neurons responding to or adding to the response with experience? We analyze actually the percentage of neurons modulated by each of the variables. So in expert animals, we see that we have around, I don't know, 40% of neurons that are modulated their activity with the context, the visual context or the position. We of course have other cells. We have leaking cell, inhalation, reward, speed, preparatory to hit, which will be the prediction of a go trial. And we can see that in naive animals or animals in the first session, we can see that really not many neurons are modulated by the context or by the go or the preparation to hit, which will be an anticipation of the go. So that is very interesting because it means that the contextual modulation of the activities actually acquire with experience. And I would like to summarize this bit of information here. So the activity in the piriform cortex is modulated by olfactory and, of course, non-olfactory variables. Modulation of the activity by context, position, and reward are acquired with experience, and the olfactory piriform cortex uses this information from other brain areas to adapt other processing depending on experience and behavior. And we have now some idea of where is this contextual information coming from. It seems that it's coming from the lateral entorhinal cortex, and we're very excited with these results. So with that, I would like to just thank the members of the lab. I mentioned some of them during the talk, but there are many others. So these are the lab members that are participating in the olfactory project, but of course we have also projects on the hippocampus and on the activity-dependent gene expression that are very important for us. So with that, I would like to thank everybody. Thank you.
Okay. Thank you so much, Antonia, for your pretty nice talk. It's very challenging and in technical aspect, and also very interesting theoretical approach. We have some questions for the audience. I just pick up the first one, and then we come back in the final remark. Francisco Estrada Rojo ask you how affect the intensity of odor the neuron response.
So hi, well, thank you for the invitation again. And so we did not try different concentrations. We are trying a pretty high concentration right now. And it's something that we would like to explore actually, how concentration of odor is actually also encoding, or how also other mixtures, for example. But we have not tried. These are very preliminary experiments that I wanted to share, but it's all unpublished. And so it's an ongoing work, and there's a lot to still do. But I would assume that you will still see the modulation of all these different behavioral variables, even if the concentration changes. I mean, we are not really focusing too much on the encoding of odors right now, but how the animal actually start to encode many other aspects of the behavior when it means something to it, right, to the animal, because when you put the animal naive, there's no modulation of other things, not much at least. But as the animals start to learn that these other aspects of the task are important to get that reward, then he starts to encode these other variables in the piriform cortex. And I believe it encodes it everywhere, probably in the brain, not only in the piriform cortex. So as the animal is involved in a task that means something to it, it will start to encode different variables in different areas of the brain, I believe. But that's something to prove.
Yeah, something we need to do. Yeah, really interesting, because especially in the olfactory system, the concentration of the odorants change your perception. So probably the changes between one context and other, you still have two different
yes,
population.
But in the piriform cortex, there is a lot of invariance with concentration. So there's a recent paper from Kevin Franks showing that there is some invariance in the concentration in the encoding in the piriform cortex. So they equalize kind of the concentration to be able to perceive the odor, right? So yes, I'm not sure if that will make a big difference in the result.
I see. Okay, thank you so much. There's many another question we will take later. Just let's move on to our third speaker of today is Professor Marcio Moraes from Brazil. Dr. Moraes is electronic engineer from the Instituto Tecnológico de Aeronáutica, and PhD in Human Physiology from Universidad de Sao Paulo. He is currently full professor at the Universidad Federal de Minas Gerais, group leader of the Nucleus of Neuroscience, and director of the Center of Technology and Research of Magnetic Resonance in Brazil. His group has been working in neurophysiology with particular interest in neural network dynamics, epilepsy, and other issues like electrophysiology. And his talk is entitled Understanding and Tampering with Temporal Patterns in Neural Network. So thank you so much, Dr. Marcio Moraes, for being here today. The screen is all yours.
My name is Marcio Moraes, and I'm a professor at the Universidade Federal de Minas Gerais. Núcleo de Neurociências is where I work, and you can find more about us if in this link right here, if you're interested. I'm very thankful for the opportunity to speak at FALAN, alongside distinguished colleagues that work within the field of neuroscience in Latin America. Well, let's get to the point. Our research interests encompass the structural and functional architecture of the brain. More specifically, how the brain encodes information within the neural networks about the world that surrounds us, and how it processes that information to output appropriate behavior. Well, I'm pretty sure I have narrowed down to the whole field of neuroscience by now. But anyhow, one way to frame this problem is to think about the 80 to 100 billion neurons in the brains in terms of graph theory. Leo and colleagues have provided us with a nice review about this subject. So the whole idea here is, suppose that I have all neurons in the brain, like 1, 2, 3, 4, up to 80 billion, represented by these nodes. So let's call these a node. And the connections between the nodes, we're going to call edges, actually shows how these neurons are interconnected. So we would actually be able to represent this by a matrix that has 80 billion columns, billion, and 80 billion rows. And each one of these elements in this matrix represent the connections between the neurons. Well, in fact, by using diffusion imaging and structural MRI, we have shown that the organization is most likely in the brain to have strong interconnectivity between groups of neurons. And these groups of neurons send projection to other groups of neurons to transfer local processing to processing pockets within distant parts of the brain. So what I'm trying to say, translating this, is if you look at this matrix right here, if every single neuron would connect to every other single neuron, I would expect this matrix to be completely full like this. But it's not. I mean, if you look at it, it actually has like pockets of information, of connections, showing that perhaps it's not that every single neuron connects to every other neuron. Well, if you think about this, it makes perfect sense. I mean, it's not so far-fetched if you imagine that the huge spatial toll that this would take, and especially metabolic toll, if you have to connect every single neuron to each other. It would be physically impossible. You would not have space to put all this white matter. So other architectures are most likely in the brain. One such arrangement is called small world networks, and it fits perfectly with the idea of modular pockets of information, transfer information to other pockets that I have just discussed. Highly interconnected pockets with distant connections among these modules through communication hubs. In fact, if you look at these small world topology, they are usually accompanied by the presence of modular structure and hub nodes, which seems to optimize both the information segregation within a specific module. But not only that, but it seems to be ideal in order to integrate information with other networks, meaning that not only it has good clustering coefficients, it clusters information in modules like this, but it also maintains the idea of short distances between these modules to transfer information among the modules. Now, if this is the case, then we are not, the brain architecture will be somewhere around here in the increasing randomness of its interconnections. Now, forgive the pun here, but if small world networks are the case, how do we transfer spatial temporal discharge patterns from one module to the next? Well, what I mean to say is, suppose that this neuron fires in this one and then this one, so in this order, and this actually is a pattern that represents something that's happening in the outside world. How do you transfer this information from module A to B? This is a big problem, and I would definitely say that we might be able to solve this problem to think about oscillations in the target module that will make certain neurons more prone to discharge at one specific time. Meaning that suppose that in a particular point of a slow wave that's passing through these neurons are with low probability of discharge, while these neurons are with high probability of discharge. So the encoding happens not where, but when this hub discharges. If it discharges at this specific point of the wave, it would probably recruit this guy. But if it discharges, I don't know, at another point of the wave, it could recruit another neuron that's more prone to discharge. So within a period of oscillation, that would be where the information of a specific neuron in module A is supposed to recruit specific neurons in module B, which means that this would be some sort of temporal multiplexing. Well, the concept of oscillations is not new in neuroscience, and actually what I'm talking about here is definitely has been proposed elsewhere. If you look at Busacchi, if you look at Varela, they actually provide, and there's a lot of evidence showing that phase encoding of specific discharge of gamma oscillations around slower oscillations like theta oscillations, they actually encode information about what the animal is about to do or specific behavioral tasks. Anyhow, how do we test this? So how do we go about and actually design an experiment that would actually be able to test this? So how do we show how temporal coding influences this dynamical connectivity between modules? Well, one idea that we had is to get a specific theta oscillation as a time frame. So this would be 100 milliseconds. So within that time frame of 100 milliseconds, we had two different patterns, both pattern A, which is this pattern, and pattern B, which is this pattern. And we tried to design a protocol that would actually get the animal to learn that pattern A, he's supposed to do something, but not with pattern B. And by applying pattern B, he would do something and not with pattern A. But in order for this not to have any idea that we were recruiting different modules or modules in different intensities, we applied the stimulation to what we thought was a good node, which is the amyloid complex. And we used this, we applied to this node the exact same amount of stimuli, so 6 stimuli in both patterns. The only thing that changed, so we applied to the same place with the same amount of stimuli, the only thing that changed is the temporal organization of these patterns. And we changed the temporal organization. So the whole idea here is we had two groups of animals, mainly. So this is the work done by Flávio Afonso Gonçalves Mourão. He's a postdoc resident at Núcleo de Neurociências. And we published this in Cerebral Cortex in 2015. But what Mourão showed is that if we had two groups, so let's call group A the group that has pattern A paired with the foot shock, and group B that has pattern B paired with the foot shock. Now, it's important to highlight that both groups received both patterns A and B. The only difference between them is that the group A had a foot shock paired with pattern B and not paired with pattern A, but not paired with pattern B. And group B had a foot shock paired with pattern B, but not paired with pattern A. But they received pattern A and B both. And the whole idea here, so this is a control only, but the whole idea here is when we tested and we applied pattern B to the amyloid complex during the retention test phase, what happened, and so this is a normal, common fear conditioning test. What we saw is that only group B actually freezed. meaning that it recognized. But when we applied pattern B to group A, since it was not paired with pattern B, it did not freeze. It did not freeze. But 24 hours after that, just to make sure, we applied now pattern A. And group B that responded so well with pattern B, when we applied pattern A, group B did not respond, or respond close to what controls would, unpaired groups would. But pattern A now, pattern A, well, this one had a lot of freezing. Just to make sure again, 24 hours after that, so 72 hours after the first test, we applied pattern B again. And when we applied pattern B again, as happened in the first day, group B had a lot of freezing and group A didn't have much freezing. Now, the whole idea here, and we tested this with other metrics too. I'm not going to get into it because of the lack of time, but the whole idea here is what neurons or what modules were actually active during that time. And if you looked at the last stimuli, we had B pattern applied. So this is everything that happened after we applied pattern B. By applying pattern B, you can see that only the B group, the one that was paired to it, actually had big recruitment of the dorsal medial hypothalamus and the ventral medial hypothalamus. Well, these are output structures to the fear behavior. So these are output pathways to the to sympatho-excitatory fear behavior. So this makes sense. But the interesting thing is the group A. So remember that group A received at this time the B pattern stimulation. It had no recruitment of the hypothalamic structures. Now, if you look at the amygdaloid complex, we saw kind of the same thing with group B, but here's the interesting data. If you look at the prefrontal cortex, no, it was only really recruited. So the prefrontal neurons or the prefrontal module was only recruited when the animal did not know that he had to have a freezing behavior. So he was like not sure what he had to do in terms of output of behavior. So in this case, you had a huge recruitment of the prefrontal cortex, which is completely consistent with what we think is the role of prefrontal cortex inhibiting the conditioned response of amygdalo hypothalamic discharge. We tried to put this idea of temporal patterns to good use, and the whole idea of to put this to good use was to use this in epilepsy. So suppose that you could think about epilepsy as a pattern of stimulation of ictal discharges. What would it take to disrupt that pattern, to disrupt those connections that recruit more and more by hyper-coupling different modules? Well, one of the ideas is perhaps we could mess up these hyperconnectivity by using different stimuli. So this is the pattern that is a discharge pattern, the normal discharge pattern that you would see in epileptic animals with PTZ-induced seizures. And this is a completely, it's a kind of a messed up temporal code. So the whole idea here is this, in what we are thinking, would actually promote or facilitate the coupling between networks, while this would disrupt it. And this is exactly what we found. We saw that the periodic stimulation that would actually resemble the firing pattern, temporal pattern of discharge of the ictus, actually was pro-convulsant, while the messed up temporal code that we used during ictogenesis actually was anti-convulsant. This is the work done by Vinícius Rosa-Cota, which is a professor now, so Professor Vinícius Rosa-Cota at the Universidade Federal de São João. He has his own research group called LINS. We actually tested this. This is Michel Bernanos. We tested this with MRI studies. And exactly the same with the periodic stimulation. Here's the amygdaloid complex highlighted. With the non-periodic, and this was quite interesting, we saw a huge recruitment of the prefrontal cortex also. But as we saw with the behavioral data, periodic stimulation increased activity in the ictogenic during the ipsilateral side of stimulation during PTZ seizures, and non-periodic blocked where the recruitment of those modules during ictogenic. So we saw that we could interfere with the coupling, but can we predict if something like that's going to happen? So I'm going to call this active probing. So in order to do that, and I'm going to go really fast by this, we had both mathematical models coupling population A and B, and we have a biological model using PTZ to couple to use for predicting. And both DBS and this mathematical model of DBS, we were actually able to show that an abnormal coupling between modules was happening. And I'm not going to have time to go into this in detail, but we actually did this both in animal research and clinical data using a sensory stimuli, that's auditory steady state stimulation, and we showed that we could actually predict. So we have pretty good indication we can predict surgery prognosis, and we actually saw a very good chance of predicting this hypercoupling in epileptic animals. Well, I'd like to thank my collaborators for this, and please feel free to contact us in using this link.
Okay. Thank you so much, Dr. Moraes, for your very nice talk. We have room from one question. We have one from the audience. So Vitor dos Santos said, from the introductory part, I had the impression the stimulation pattern were nested in ongoing theta cycles. Is this the case? If so, have you tried to make it uncoupled to theta?
Well, thank you very much, Vitor. That's a very good question. And yes, that's exactly what we tried to do. We looked at specific slow wave oscillations and tried to fit our pattern into one of these pockets of information. And to answer your question, well, yeah, ongoing research, we're actually trying to change this in such a way that we're looking at different periods of oscillations. And the difficulty here to do what you're asking us to do is that I will have to do an online processing and actually get the theta wave as it's happening and inject the pattern in the animal as I'm recording the theta waves. But yeah, it's possible, and I think we actually solved that technical problem now, and I hope to have the results out of this coming soon to show you.
Nice. Well, thank you so much. We hope to see this results pretty soon, really. Okay, there are another question we will take later in the final remark. Let's move on for our last speaker of today. Thank you so much, Dr. Moraes. So our last speaker is Professor Xiaoyu Xu from Chile. Dr. Xu got her bachelor in neuroscience from New York University and her PhD in neuroscience from the University of Rochester at the US. Currently, she is associate professor at the Instituto de Neurociencias, Universidad de Valparaíso in Chile, and in the Centro Interdisciplinario de Neurociencia de Valparaíso, Max Planck Tandem Research Group, leader at the Universidad de Valparaíso. Her group has been working on the regulatory mechanisms of GABAergic synapses and their impact on neuronal functioning. And her talk today is entitled Diverse Regulation of GABAergic synaptic transmission and plasticity. So thank you so much, Dr. Xiaoyu Xu, for being here today. And the screen is all yours.
Hi, welcome back to FALAN Networking 2021. In this talk, I would like to share with you two projects that reveal a diversity by which GABAergic inhibition can be regulated. We know that neuronal activity shapes brain function, and this is exemplified by this video of calcium imaging in the visual cortex of mouse that's viewing visual stimuli. You can appreciate is an increase in response, an increase in neuronal activity in response to stimulation. And if we were to look at any given neuron in the visual cortex, what we find are, in addition to these action potentials that are revealed by the calcium signaling, there are also subthreshold events that shape these action potential output. And they are actually the activation of synapses along the neuronal dendritic tree. Okay, and there are two forms. There are depolarizing events that are mediated by glutamate from glutamate producing cells, and inhibitory events that are mediated by GABA released from GABA producing cells. Generally, neuronal communication is thought to be driven by excitatory transmission because there are many more excitatory cells and they make the majority of projections across brain regions. But we know that inhibition is important for information processing because when inhibition dysfunctions can trigger neurological, neurodegenerative, neuropsychiatric disorders. Through decades of work, we know that inhibition influences brain function at multiple levels, including behavior, activity across brain regions, and in cells and synapses. So changes in inhibition can have profound effects on overall brain function at multiple levels. But it's unclear precisely how inhibition regulates all these behaviors. And that's because at least one of the main reasons is there's a rich diversity of GABA producing cells. In the cortex, there are three main non-overlapping types that can be identified by whether they co-express pyroalbumin, somatostatin, or the ionic serotonergic receptor. Okay, there are many differences in the interneurons. One key difference is in the circuit that they make. Okay, so PV synapses tend to affect the spike timing of pyramidal cells, whereas the other interneuron types inhibit the dendrites where excitatory synapses occur. Okay, VIP cells, which is a subpopulation of the serotonergic subtype, actually also mediate disinhibition because they inhibit inhibitory cells. So one of the key questions in my lab is to understand how neuronal activity can differentially regulate these diverse forms of inhibition. Because you can see changes in these different sites can have different effect on how the pyramidal cell will compute the input and transform it into an output. We are particularly interested in glutamatergic influences because a balance of excitation and inhibition is important for normal brain function. So in our first study, what we examined is the effect of activation of NMDA receptors on diverse GABAergic synapses. And that's because NMDA receptors are a key subtype of glutamate receptors, and they also play key roles in multiple forms of plasticity at excitatory synapses and some unidentified GABAergic synapses as well. So to address this question, we need a way to selectively activate different populations of GABA cells. Okay, and here we take advantage of optogenetics. All of you are probably familiar with it. We basically take advantage of the ability of light to open channel rhodopsin. And if you conditionally express these channels into specific interneurons using a Cree recombinase strategy, we can selectively activate these interneurons in acute brain slices. Okay. So we first looked at the ability of NMDA receptor activation to affect inhibition from somatostatin interneurons. Okay, so when we patch a layer 2-3 pyramidal cell, obtain some baseline iPSCs, evoked by activating the some interneurons that express channel rhodopsin, we saw a profound increase in iPSC amplitude. Okay. And this is not observed when we looked at iPSCs mediated by activating PV interneurons nor VIP interneurons. Okay. And we were interested in the mechanisms of this LTP of inhibition. And through a series of pharmacological manipulations, we determined that NMDA receptors in the postsynaptic cell that we patch triggers calcium increase that increases the activity of CAM kinase, and that leads to an increase in GABA A receptor number. Okay, we wondered what is the mechanism for this differential sensitivity of this synapse to NMDA activation compared to those mediated by PV interneurons or VIP. Could it be the location of the NMDA receptor? So we directly activated CAM kinase and bypassed NMDA receptor activation using by loading calcium and calmodulin into the pyramidal cell. So if NMDA receptor location is setting the difference in the ability of these different inhibitory synapses to be potentiated, then this manipulation should increase inhibition from PV interneurons. But this is not what we saw. So inhibition from PV interneurons remains steady throughout the time course when the CAM kinase will be activated. But instead, the somatostatin interneurons, the inhibition mediated by somatostatin interneurons increased. And this increase is dependent on CAM kinase activation because it's blocked by the antagonist KN62. So something is special in the signaling pathway after CAM kinase activation or in the NMDA receptors themselves. We do know that GABA receptors are actually very heterogeneous. They're made up of different composition, subunit composition. Okay, so they're pentameric, made up of 2 alpha, 2 beta, and a gamma at synapses. But the alpha subunits and the beta subunits can vary. And there's several subtypes that have been shown to be able to be phosphorylated by CAM kinase. We particularly were interested in the beta 2,3 containing GABA A receptors because we found in experiments that I will not describe an enrichment of these GABA receptors at GABAergic synapses from somatostatin interneurons. Okay, but do they play a role in plasticity? So we took advantage of conditional mice where we can, with Cree, delete the expression of GABA A receptors that contain beta 2. Okay, and by doing that, we can abolish the LTP triggered by NMDA compared to glutamate controls that still express the GABA receptors with beta 2 subunit. This is not seen with a conditional knockout that we made with beta 3 containing GABA receptors. So the LTP that we see is probably due to CAM kinase directly phosphorylating beta 2 GABA A receptors. Okay, and so far I've discussed a mechanism for inducing plasticity at somatostatin interneuron mediated synapses, right, but not other GABAergic synapses due to a difference in the GABA A receptors at the synapse. Now I'd like to switch gears a little bit and talk about tonic inhibition. Okay. So the difference between tonic inhibition and synaptic inhibition is in the time course. So synaptic inhibition mediates these fast synaptic events. Okay, tonic inhibition, as you can tell from the name, the receptors are tonically active. So you can see the effect of tonic inhibition by blocking these receptors, okay, with gabazine, one antagonist as an example. So this project actually started from an initial project that we performed showing that somatostatin interneuron can suppress calcium signals in dendritic spines. Okay, and this is by combining optogenetics with two-photon calcium imaging. And so what we found first, this is shown in the video, is an increase of calcium signals that invades the dendrite and the spine, here labeled in red, as a result of an action potential triggered in the axon initial segment. Okay, and this is due to the activation of voltage-gated calcium channels. What we found is when we activate somatostatin inhibition, we can reduce the calcium increase triggered by the back-propagating action potential. Okay, but we unexpectedly found that blocking GABA A receptors, in addition to preventing the inhibition achieved by the somatostatin interneurons, it also reduces the calcium rise due to the back-propagating action potential themselves. Okay, and that's shown here compared to if you just apply control solution. So since it's unclear how inhibition can lead to an increase in calcium signal, we turn to our collaborator, Tom, who made a two-compartment neuronal model, right, with a soma and a dendritic segment. And he put in the normal conductances for neurons and added a conductance for GABA and conductance for the two types of voltage-gated calcium channels, the two big subtypes of voltage-gated calcium channels. And when he simulated an action potential, what he found was when you block the GABA conductance, it triggers a depolarization of the membrane potential in the dendrite, not in the soma, and thereby reducing the calcium current triggered by the action potential. And this effect preferentially affects the low voltage gated calcium channels and not the high voltage gated calcium channels. So we wanted to see if we could mimic this experimentally, and we took advantage of selective antagonists for the low voltage activated calcium channel, the T-type, using TTA, and to compare that with the effect of a cocktail of antagonists for the high voltage gated calcium channels. So both types of calcium channels contribute to the calcium that is triggered by back-propagating action potentials to an equal amount. But only when we block the T-type calcium channels do we block the depression induced by gabazine or any other GABA A receptor antagonist. The blockage of high voltage gated calcium channels do not have an effect on gabazine induced depression of calcium signals in the spines. Okay, so tonic inhibition facilitates calcium signaling probably through T-type calcium channels. Okay, and so does this effect have some kind of physiological function? We took advantage of a short-term form of synaptic plasticity that is calcium dependent. Okay, so calcium triggers the production of endocannabinoids that then can suppress GABA release. Okay, so when we induce a burst of action potentials that leads to an increase in calcium, you can see a transient suppression of inhibition that recovers within a minute. When we apply a low concentration of gabazine that reduces calcium triggered by the burst of action potentials, but does not block synaptic inhibition, we can see that it blocks the short-term depression of inhibition. Okay, so suggesting that tonic inhibition can facilitate the expression of the short-term form of endocannabinoid depression. Okay, And so this comes to the end of my talk. Just like to highlight the diverse mechanisms of regulating GABA synapses, one through NMDA activation that selectively increase the number of beta-2 GABA receptors in synapses that are mediated by somatostatin interneurons. And then there's a population of extrasynaptic GABA-A receptors, probably alpha-5 containing GABA receptors, that enables the activation of T-type calcium channels, which then can trigger increase in calcium rise to lead to a transient suppression of inhibition. Okay, and with that, I'd just like to thank my lab members, collaborators in the US, and my funding sources. Thank you.
Okay, thanks so much, Dr. Shayu. Very, very nice talks. So that remember me to usually when we talk about plasticity or LTP phenomena, everybody thinking in glutamate and MDA receptors, and we forgot the GABA cells. So it's tremendous interesting that you show it today. So let me see, there are some questions from the audience. Still not yet, but I'm just wondering, what about another GABAergic neuron that neurons usually have really important for oscillation process, like ripples in the hippocampus that is really important for memory consolidation, for example. Do you think it's similar mechanisms mediated by calcium, or you have another possibilities?
Right. So there are even forms of LTP that occurs at PV interneurons. The most common one that you see is something that's dependent on the action potential firing of the pyramidal cell. So it seems like there is some kind of segregation between PV interneurons that shape action potential firing, and then the action potential firing can modulate their inhibition. And that's kind of a separate mechanism from the dendritic inhibitory synapses that shapes excitatory synaptic transmission, but on the other side, excitatory synaptic transmission also shape them. So that's a nice kind of way to sort of self-regulate their own function. So there are processes like that. The general one that's observed when they've been characterized, right? So traditionally people don't distinguish interneuron types. It kind of, because it was hard until optogenetics came along, right, to really do it. But the more recent work show more of a presynaptic mechanism. So it triggers NOS to be released is one example in the cortex that then travels back into the terminals of the PV interneurons. And then they increase GABA release. So the mechanisms of how these processes are triggered are different as well.
Yeah, sure. That's really interesting that every kind of interneuron can affect differentially the action potential.
Right. And I think that's one of the reasons why there's such a high diversity, right? Because they all have kind of their own different functions. And to really regulate their function, they need a very specific stimuli or signal. You know, you don't want all inhibition to increase or decrease in the same way.
Exactly. Okay. That's nice. So, well, it has been a real pleasure to be today with all of you. So right now we have space, we have room for final remark. We will take a round of questions for all of you from the audience, and there are others here. Let me go back to Professor Zamengo. Ines, there are some questions for you that leave behind. So Antonia Marie Burgen asked you, let's say it's a great talk, and they ask you, how plastic is this phenomenon? Does the experience change it or not? What do you think?
How plastic is the metric, you mean?
Yeah, I think so.
Good question. We have not checked for that. I mean, we have checked for plasticity in the metrics of remembered colors, and we didn't see much of an effect. But we should still check for the perceptual metric. I mean, it's not the same to determine is the first color that you can distinguish perceptually than to distinguish the first color that you can distinguish mnemonically with your memory. You can try to remember colors in specific sets, right? On the other hand, I'm reluctant to say there is no plasticity, because that's almost kind of against to what I would expect, but we didn't see it. So we did not observe plasticity in the first detectable color through our training. I mean, we are the most trained subjects of the experiment, of course, because we've done these experiments time and time and again, right? And perhaps in the first few trials you become better, but then you very fastly you reach some sort of plateau where your discrimination ability kind of saturates in a way. That's what we have seen, but we didn't check specifically for that. I have to say that. So that's something to come in the future.
Yeah, thank you so much. So a lot of food. That's good. We have another question for Professor Antonia. Grace Moraes asked Antonia, as you suggestion that piriform cortex codify a space like play cells in the hippocampus? Interesting question.
Well, yeah, that's a great question, and I had it before. Well, it might be, you know, and we are not the only ones seeing this. So there is also some very new data from Cindy Poo in a different task. And she also observed some kind of place representation in the piriform cortex. But my belief is that really what the piriform cortex is encoding is really what matters for the animal at that particular moment. And if it is a spatial-oriented task, because the animal has to learn, for example, where the odor is, or yeah, where in the corridor in our case, but if it's a mouse running through the environment, he also has to learn, is this odor coming from here, and that would predict some reward, for example. Then the space matters, and then probably the space is represented not only in the hippocampus, like you would always think, but it's also translated to other areas, because now the space means something related to, for example, an odor, or if it's somatosensory, would say it can also maybe represent the space. And so recently some people have seen in the visual cortex that they also can find, but the visual, again, visual and space is harder to separate. It's not like the others, but they see resemblance of place fields like in the hippocampus, also in the visual cortex. So I think it is recently been more thinking of that the sensory areas are not just purely representing the physical aspects of the sensory stimuli, but it's also representing other aspects of behavioral aspects of the animal too. And one could be also contextual, and the contextual, of course, is probably coming from the hippocampus. And very recently we did an experiment turning off the entorhinal cortex while the animal is doing this task, and the animal starts to confuse the context. So they just do right with the others, but they don't do right with the context. So we know that the signal is coming probably from the hippocampus through the entorhinal cortex. But this is, we only did it behaviorally. We need to now check if the cells now lose this modulation by context.
Thank you so much. Really interesting. So it's not weird that you can find activity like play cell like in another brain regions. I remember in the medial prefrontal cortex there are some cells that show Bruno Pousset a long time ago that they have this spatial information related with reward. There are something interesting. The neuron
because
do something.
The reward is marking that's
exactly.
important.
Yeah, the reward is what is changing the activity of these neurons. Very nice. So we have now another question for Professor Moraes. Vinicius Rosa Cota asks you, do you know if a small world network topology deal with power law phenomena?
Yeah, well, I already answered him in the chat.
Yeah.
But yes, it does. It does relate to that. It's a very optimized way of conveying information from one part of the brain to another part of the brain. And I would definitely say that it makes it very efficient in terms of energy, metabolism, and makes it very efficient in terms of the organization of the system. So yeah, the answer to that question is yes.
It's yes, that's it. Good. Doctor, okay, I have another question for Professor Chayushu. It's during Marcia Moraes, this is her great talk. Did you look? So he asked you, did you look at CCK GABAergic neurons related to the endocannabinoid system?
No, I didn't. The short answer is no. It's tricky with so you know they have CCK3 mice. The problem with them is that because it's CCK is expressed by pyramidal cells earlier in development, so you know we have to come up with a trick to basically not express them in pyramidal cells and only in the CCK interneurons. But interesting you should say that, I didn't talk about this data, but somatostatin interneurons also have CB1 receptors in their terminals. And they show plasticity that's endocannabinoid dependent.
The reason, Can I go ahead and interfere just 1 second? Because I mean, since I'm here, would that be okay?
Please go ahead.
Well, we wrote a big review right now, and in terms of the endocannabinoid system and its role, not only as a circuit breaker, but actually in synchronizing different portions of the hippocampus. And of course, this applies directly to therapeutic strategies in epilepsy. And the importance of the CCK neurons, much more than the PV neurons, exactly the same that you showed, seems to have a more important role in synchronizing networks that are kind of far away apart. And I think this is the exact same thing that you showed in the results, right? So
yeah.
For
so
the
interesting
thing, so.
Right. And so maybe so in the hippocampus, CCK are basket cells and they're also dendrite targeting. So I don't know if it's true or not, like maybe that more far distributed regulation is from its dendritic influence. But yeah, I haven't looked. It's something I definitely thought to do.
Work to do, yeah. Let's move on again. So we have another question from Professor Zamengo. This say Rodrigo Laje say, hi Inés. Does pure influence the geometry in color space? An example, knowing that the object should be in a given different color, etc.
Again, I think I would frame it in the same way that I replied to Antonia. Hi, Rodrigo, first of all. I mean, of course, if you know that there should be something, maybe you are more careful in looking at it, so you pay more attention, so to speak. But our experiments are, we have two types of experiments, right? On the first type of experiment, you have to detect a difference. So there, I mean, half of the time there will be no difference, and half of the time there will be a very small difference. So the experimental setup doesn't allow you to know in advance whether there will be a difference or not, because that's what we want to test, so to speak, right? And in the second kind of experiments where we worked with chromatic induction, when you are adjusting one color surrounded by, say, one context in order to match different colors surrounded by a different context, of course you know that when you see them equal, they are not really equal because you're compensating for the context, but you can't help it. Even if you know that that is the situation, you may want to say, I want to trick the experimentalist, and I will put something that I don't see. Okay, you can do that, right? But you can't help it. I mean, you see the effect of induction. It's a very strong effect. I mean, and we're all kind of familiar with that, right? We can't help seeing, responding to borders of contrast in the same way we cannot help seeing chromatic borders. I mean, it happens. If you know, you will perhaps know in which direction you're being induced, so to speak, but that won't take the effect away.
Yeah, that's true. Good, thank you. So, yeah, I was thinking in my experience in your image. So you told me that some figure in the background must be see it yellowish or something like that. But there are some pair of stimuli that even see it, because as you know, I'm blind for such color, and even though you tell me what I have to see,
okay,
it doesn't change very much.
You didn't see.
Yeah,
but some of them you should have seen. Maybe not all of them, but some of them you should have seen, right?
Yeah, some of them. Yeah.
Okay.
Yeah. There are, I think, in this row and columns, there are at least 4 pairs that I didn't see it.
Okay. I fully understand that. Yes. That's the way it should work. If you don't see any difference at all, then maybe you're very severe colorblind.
There are some that I cannot tell you which is the color, and there are a couple that I didn't see anything. There are 2 of them. Really interesting. So you just send me the test, please. Okay, we have another several question for Antonia. Enrique Pontescay asks you, what kind of odors do you use? Are there stimular or depressing odors?
Yeah, no, we're using monomolecular odors that are not, doesn't mean in principle anything to the mouse. So we're not using, like, I don't know, fog smell or urine smell or things that innately cause something. So we want something that is not, doesn't have any experience before with it, because we want to check how experience changed the representation. But I'm sure if you use odors that have some previous experience or have an innate behavior, that might change, of course, the representation. I'm sure. I mean, that, I don't know. It might be that you can also associate that with a context, but still it has some other issues already from the beginning. So we wouldn't probably see the change we see from naive animals to experienced animals.
Okay, that's good. So the odor is not highly arousing for the animal.
No, the odor is it's a new odor. It's a new odor. Yeah, it's neutral in terms of the behavior.
In terms of the okay.
Yeah,
that's nice. Oh, Chayu, Professor Chayu, have another question from you here. So, do you know whether this learning happens without the rewarding contingency? So, in another word, is exposure to the others or visual context sufficient for that?
Well, we didn't try that, but I mean, the way we motivate the animals to learn the association is with the reward, is water, because they are thirsty. We didn't want to do it with a punishment, because you can also do a fear. We wanted to be a positive reward, but I don't know. It means it has to mean something for the animal. I think it has to want to learn, and then learn it because he wants to drink, or he wants. If it's just foraging without really having a goal, I'm not sure how much that will be represented then in the, for example, piriform cortex. But I don't know. We haven't tried. We just did it with the reward. Yeah. But I think the reward is an important signal. It's a very important signal that probably kind of labels that this circuit has to be potentiated, for example. Yeah. So that's what I think. I think the reward marks that the input that are arriving at that particular moment when there is a reward going on will get potentiated, and then will be expressed in the population of neurons that are responding to that input. For example, the input coming from the entorhinal cortex that could confer the contextual information. Maybe that input is always there, because of course it has to arrive to the piriform cortex to actually mean something at the beginning, but then it only gets potentiated, and then cells are responding to that input when it's associated with probably a reward or a fear. Yeah, so something has to be there to tell the cortex, okay, this input is important, and it's to get represented there together with the other. Yeah. So I think that's either reward or punishment, or maybe, I don't know, some other type of stimuli that are very important for the animal.
May I add a question to Antonia?
Sure.
Now she's here.
Sure, please.
What's the role of active running? I mean,
well, that was actually the way we found to have a head fixed animal, but that goes through different contexts. Because we wanted to head fixed, because we needed to record from the animals acutely.
Okay.
And that's the way. But having said that, we did see a modulation with speed on the cells. So normally cells tend to decrease their firing with speed. So the filter that we obtain for speed in the GLMs, it's a filter that decreases with the speed. So normally cells that, when the animal goes slowly, has a higher firing rate than when the animal is running. It could be related to attention too, because when it's running fast, it's probably not attending too much.
Can it be related also to ventilation or to breathing speed?
So we know, because we do measure inhalation rate, and we uncouple those two. So actually also inhalation rate has a lot of modulation on the response of the piriform cortex and probably other areas of the brain. And it's interesting that behaviorally, when the animal learns that the odor is coming, it already starts to increase its inhalation rate. It's a predictive thing. So we need to uncouple sometimes these things. That's why we're using GLMs to know exactly what the neuron is responding to, because there's so many things that are going on when the animal is running.
No, and I know that, for example, play cells, the grid cells, do not show up if you have the animal transported, right? Yeah, you need them. No, even if you put them on a car and take them, it doesn't work. You need them to be. So I was wondering whether
they are moving.
Yeah, I don't quite know. That's why I asked.
Yeah, I don't know if it's related to really the pattern generator of going really moving itself, or when you move it, he doesn't really feel that he's really moving.
Or maybe the theta rhythm has another amplitude or something like that.
Sure. Yeah, maybe it's related to that. Yeah.
Yeah. Thank you so much, Dr. Zamengo and Dr. Marin. So it's a really interesting issue. There are some people working in active sensing today, where the movement during perception is really important. So when you are smelling, you are moving your whisker, or when you are seeing, you are moving your eye, etc. And there are a percentage of the variability of the response of our neuron that is command for movement. So there are a window to explore this kind of thing also.
Yeah, absolutely.
Yeah, thank you so much. I have another question for Professor Marcio Moraes. So Tamara Bustamante asked you, Dr. Moraes, great talk. She asked, for how long is non-periodic periodic electric stimulation administered in animal with chronic epilepsy? Is this a daily basis or protocol?
This is a very good question, because she's thinking about clinical applications, and we actually have some of these trials almost ready for clinical trials right now. So we actually starting to do this in the hospital. But the idea here is the models that I've showed so far, I know when the seizure is about to happen, because you're not like a spontaneously recurring seizures. They are seizures induced by drug. So I actually put the drug, and I know that the circuit would actually come to an unstable form of functioning and have discharge patterns in a few minutes. But just not to leave that question completely unanswered, we do have models that have spontaneous seizures. And we had some really, really good controls, results using at least 2 stimuli a day, one in the morning, one in the late evening. But what we really are trying to aim at is to join both parts of the lecture. So meaning you detect that the circuits are being hypercoupled. So you detect that somehow information from A is abnormally transferring to B, and this should not be happening. When that happens, then you apply the non-periodic stimulation. So we call this a closed loop treatment. So you detect the seizure, and you only apply the stimuli when you think a seizure is about to happen. Because the problem of applying this throughout a long period of time is that it obviously affects other functional networks. So if you use like a depressant drug or phenobarbital in a patient with epilepsy, what happens is that he's going to be sleepy, and he's not going to perform cognitively so well. And this happens because you affect the baseline of what we call the functional connectogram of the brain. So the whole idea here is not to apply it the whole time. I mean, that's exactly what we're aiming at, is to apply it only when it's needed, only when the circuits show signs of being, of moving towards instability. So that's why we need the detection process working together with the NPS stimulation. But yeah, okay, we did that, and it does work. It does reduce seizures if you apply it twice a day. We didn't test it lower than that, but twice a day in animal models of epilepsy that have spontaneously recurrent seizures.
Thank you so much, Marcio. Really interesting issue about that. Especially we are thinking to do some apply method for epilepsy. Yeah, need to know.
We actually defended the Sergio Cansado, which is a neurosurgeon. We're doing implants, deep electrode implants in the hospital here in Belo Horizonte. So this is Belo Horizonte. People that only know Brazil through Nordeste and Rio de Janeiro and São Paulo. Yeah, this is a beautiful place to visit. You people should come. But the hospital here is actually doing these deep electrode implants, and we are using this for seizure detection, or at least prognosis of surgery, of specific removal of parts of the brain that we think are where the epileptic foci are. So yeah, we're moving close to actually doing the clinical trials for that.
Nice, pretty nice. So we are close to the time, Daniel, but I just want to take the last question for Professor Chayu and close this symposium. There are a lot of other questions. It's really, really interesting, all the topic and the convergency of all of them. in how the brain works, and that's the point. So to end this round of questions, Professor Chu, there are a question from Antonio Marin. This is a great talk. For the first part, do you think that the difference in plasticity between parvalbumina and SOM is related to different function of the inhibition they provide, or not?
Well, I hesitate to say related, right? Because I kind of suggest almost causal, like a cause, and what I would say is more, at least it's set up. It's the mechanisms are set up so that you can have really effective regulation of its function, right? And that could be, who knows, it could be just incidental. It happened somewhere in evolution and it worked out well, so it just propagated. But that's what you have is and there are, because they happen at different sites, right? And you can regulate the molecular.
I can, since I'm here, I was thinking more in the dendritic versus somatic inhibition, that one is more, I don't know, plastic than the other, maybe, because one actually modulates the inputs the cell receives, and the other one modulates whether it's going to spike or not. So that's why I was thinking, do you think that's why maybe one is more plastic than the other?
I wouldn't say one is more plastic. I just think the way they can be induced are different. One that requires maybe more firing, and that it could be related to just where the synapses are also, right? They are closer to where the soma is. But the interesting question to me is always when you have more complex situations, right? When you have glutamate release and you have axonal firing, somatic firing, which is what normally happens, then how do you regulate inhibitory synapses? What's the sensitivity and the threshold and that kind of thing? But there's definitely a relationship just by the location and the temporal, the timing, right? The coincidence of things. Yeah.
Okay, thank you so much. Well, we are on time. For me, I follow all the rest of the afternoon, but we need a program. Thanks so much to every one of you. This was an amazing symposium. I really enjoyed it. So, Danielle, I think we are ready. Okay.
Thank you so much.
Thank you so much. So everyone,
Thanks for the opportunity.
see you. Yeah.
Great to see you.
Yeah.